Neuropeptide Y promotes adipogenesis in chicken adipose cells in vitro. Neuropeptide Y is a neurotransmitter that is a 36 amino acid peptide chain and is found in both the central nervous system and the periphery. Neuropeptide Y was first discovered in a pig brain in 1982 by Tadimoto, and 2 years later was discovered to be an orexigenic factor in mammals through research conducted by Clark, Levine, and Morley. 3 years later, in 1987, Kunzel discovered it to be an orexigenic factor in chickens as well.
Through knockdown studies, neuropeptide Y was shown to play a role in adipogenesis in mammalian models. Zhang observed neuropeptide Y in visceral adipose tissue and also that neuropeptide Y proliferated pre-adipocytes in mice. In conjunction, these studies show that neuropeptide Y promotes energy storage in adipose tissue and inhibits brown fat activation through the central and peripheral pathways in mammals. Chicken adipogenesis is less known compared to adipogenesis in mammals. The broiler chicken is very important to the agricultural industry, and due to the continued selection for body weight and feed efficiency, the ability to control their body fat has become an important issue in the industry. The body fat accumulation of broiler chickens may also help us to understand the mechanisms that lead to obesity in humans. It is known that the central injection of neuropeptide Y in chickens causes them to start eating. However, it is unclear what the effects of neuropeptide Y are on chickens' adipose tissue function.
So, why is there a need for this research? All of these studies on adipogenesis have been on mammals. So far, there have been no reports on the effects of neuropeptide Y on chicken adipose tissue function. Therefore, the objective of this study is to explore the effects of neuropeptide Y on adipogenesis in avian species using the broiler chickens as the model. Our hypothesis is the neuropeptide Y system plays a role in lipid accumulation in the adipose tissue of chickens. The cells we used came from abdominal fat from 14-day-old broiler chickens. In preparation for the experiment, primary adipose cell culture was minced, blood cells were removed, then the tissue was fixed. Tissue was incubated for 48 hours prior to experimentation. Tissues collected were triplicated from each chick used for experimentation. Experiments were run to examine cellular proliferation, adipocyte differentiation, glycerol-3 phosphate dehydrogenase activity, and RNA isolation. Fetal bovine serum was used for the proliferation experiments because in our preliminary experiments, chicken serum inclusion was associated with spontaneous differentiation into adipocytes that confounded effects on proliferation.
So what were the results? Figure 1 demonstrates the results of neuropeptide Y exposure during the first 4 hours post-treatment. mRNA abundance was not affected at 4 hours post-treatment in any of the following proliferation markers: GATA-binding protein 2, Ki67, Kruper-like factor 1, and Kruper-like factor 7, hence their lack of statistical significance in Figure 1. In comparison to control cells at 10 nanomoles, neuropeptide Y receptor subtype 2 has an increase in mRNA abundance. In comparison to control cells at 100 nanomoles of neuropeptide Y supplementation, topoisomerase 2 alpha and thyroxidine-dependent peroxidase 2 mRNA abundance increased, while MPY expression increased. In sum, there was no effect in MPY on cellular proliferation during the first 12 hours post-treatment in cells that were induced to proliferate, and there was only a change in abundance of mRNA for some proliferation markers at 4 hours post-treatment. There were no effects displayed day 2 post-differentiation. By day 4, cells clearly showed brown adipose lipid accumulation. By day 6, glycerol-3 phosphate dehydrogenase activity increased and displayed statistical significance at 100 nanomoles of neuropeptide Y supplementation, as shown with the control group being labeled B, and the only concentration with a different label A being that of 100 nanomoles. Within these images, you can visualize the significant difference in the lipid accumulation in day 6 post-induction of differentiation from the treatment of different doses of neuropeptide Y compared with day 2 post-differentiation that was charted in Figure 2 on the previous slide. This is demonstrated by greater abundance of the red-stained adipose cells in day 6 in the 100 nanomole concentration compared with a control. Figure 4 shows that neuropeptide Y affected the expression of each protein during at least one period of time expected for cat enhancer binding protein beta, which was not affected by any dose during any period of time. The fatty acid binding protein 4 mRNA abundance increased on days 2 and 6 post-differentiation. Topoisomerase 2-alpha mRNA displayed increased quantities in response to neuropeptide Y treatment at days 2, 4, and 6 in comparison to the control group of cells that were not treated with neuropeptide Y. Thioredoxin-dependent peroxidase 2 mRNA displayed increased quantities at days 4 and 6. GATA2 mRNA displayed increased quantities at days 2 and 6. In contrast, Kripl-like factor 7 expression decreased with neuropeptide Y supplementation solely at day 4 post-differentiation compared to its control group. Overall, most preadipocyte and proliferation markers increased post-differentiation.
So what do these findings mean? Since we based our doses of neuropeptide Y on doses used in similar experiments done on rodents, we thought we would observe similar results, but we could not associate neuropeptide Y with increasing expressions of preadipocytes or proliferation biomarkers. One reason may be that 4 and 12 hours wasn't enough time for neuropeptide Y to properly impose its effects on the cell genes. We chose these times due to the short half-life of neuropeptide Y and wanted to make sure that normal proliferation of untreated cells did not dilute the early effects of neuropeptide Y. We are unclear why the proliferation biomarkers were less expressed when treated with the highest doses of neuropeptide Y. Prior to deciding on which method to use for this study, we did a comparison of previously completed experiments results reported to choose the best way to examine adipocyte differentiation in broiler chickens. Chicken serum was ultimately selected as the preferred culture media because there is a greater response in adipocyte differentiation compared when cultured with fetal bovine serum. Titrations were run to reduce confounds, and the optimal dilution for reducing these effects while maintaining effective neuropeptide Y differentiation analysis was found to be 2.5%. As reported previously in Kuo's experiment, neuropeptide Y function can mimic that of insulin. Therefore, titrations were also run on insulin to dilute it for examination of neuropeptide Y without insulin confounds. While neuropeptide Y in the higher concentration was associated with changes in adipogenesis-associated factors, the most significant effects were observed 6 days post-induction of differentiation. Using the findings of Swerzinski et al. 2003 and Sledzinski et al. 2013, we know that glycerol-3 phosphate dehydrogenase is strongly associated as a marker of adipogenesis due to the low activity of kinase in adipose tissue and production of glycerol-3 phosphate. Knowing this supplements the indication that there is a positive correlation of G3-PDH with differentiation of precursor cells into terminally differentiated adipocytes. This is driven by the mechanism that catalyzes G3-PDH into G3P, which acts as the primary component for fatty acid to lose an alcohol group during TAG synthesis. Since we observed greater lipid accumulation, we can infer that neuropeptide Y increased the rates of lipid accumulation at 6 days post-differentiation.
There was uncertainty if there was a feedback effect with regards to neuropeptide Y gene expression level from neuropeptide Y-induced differentiation in chickens due to limited research availability. This question was raised from abundant data from mammalian models. Additionally, it was unknown if the transcription factors involved in the expression of genes that increase the rate of differentiation in chickens have early, intermediate, or late gene expression reducing effects in adipocyte differentiation. Findings then showed that neuropeptide Y supplementation caused an increase in neuropeptide Y expression at days 2 and 6 post-differentiation, which also caused a 10-fold increase in expression of 100 nanomole neuropeptide Y treated cells. This answered the uncertainty, suggesting there is a positive feedback effect of neuropeptide Y causing expression of itself in adipose cells. In summary, the experimental data and findings suggest that neuropeptide Y might promote adipogenesis in chickens through 4 methods: neuropeptide Y receptor subtype 2 mediated induction of preadipocyte proliferation, fatty acid metabolism, transport, and tag synthesis. When purchasing broiler chickens as test subjects, drawing from a single company limits the genetic variation, which is not a good model for avian species in general because it makes the results more specific to that particular broiler chicken line. For more accurate data, a variety of avian species need to be used for further experimentation before making conclusions about effect of adipogenesis from NPY supplementation in avian species in general. Additionally, why is the chicken model more attractive than the mouse model in understanding obesity when the mouse is mammalian and more closely related to humans than are chickens? A chicken model makes for a less effective comparison than does a mouse model.